Hello everyone. Today's topic is mechanism of enzyme catalysis. In this topic, we are going to learn about enzyme active site, activation energy, and the formation of enzyme substrate complex. And we are going to learn about two important theories. Those are lock and key theory and induced fit theory, and four catalytic mechanisms, which includes catalysis by proximity, acid-base catalysis, catalysis by strain, and covalent catalysis. So before going to the main topic, so we need to learn about the active site of enzyme. So this is the active site of enzyme. So here this active site of enzyme, so which is flexible in nature. So why? Because it is undergoing some conformational changes, and it has two important sites. So this is the binding site. Here the substrate molecules will be attached, and this is the buttressing site, so which is also called to be catalytic site. So here the catalysis process will be occurred. So this is the substrate. Here it is attaching with the active site and forming enzyme substrate complex. Here the substrate, so which is complementary to the site of active site. Initially it is not complementary, but when substrate is attached to the active site, here the enzyme area active site will be complementary to the substrate. So that means it is undergoing some conformational changes. So when the substrate is attached at the active site. The next one is activation energies. So here in this activation energies, we are going to learn about the importance of enzymes. So how this activation energy is important for the catalysis of reactions. So here we are having three important cases. So in the first case, here there is no involvement of any enzyme. So this is uncatalyzed reaction. So here we are taking substrate as a substrate. So this substrate is converting into products. Here the metal stick we are taking as a substrate here, and here it has three important stages. So first one is reactants and transition stage, and third one is product stage. So these stages here we can see in this graph. So this is reactant stage and this is a transition stage. The peak of this area is called transition stage, and this is the product state. And here we can see another two important graphs here. If you are considering this enzyme is having a complementary shape with the substrate. So if you are comparing with this first case, here enzyme is consuming 100% of energy to reach its top to the transitional state and converting into products. But here the enzyme will be helping to the substrate uh, to reach the top of the that means to reach the transition state and converting into products. So that's why here, so 50% of energy is consumed by the enzyme, and 50% of energy is consumed by the substrate. So substrate energy will be comparatively less with the comparing with the uncatalyzed reaction. But in the third case here, the active energy, the activation energy is very less in comparing with these two graphs. So why? Because here the enzyme is not complementary to the substrate. So here initially, so there is in complementary position will be occurred in between these two, and here at transitional state, so this substrate will be attaching completely, and here so this enzyme um, uh, will be forcing some maintaining some strain on these substrates and converting into products. So here 75% of energy will be expended by this enzyme to convert the substrate into products. Only 25% of energy so will be consumed by so these substrate molecules. So that's why here the activation energy will be very less. So when it is comparing with the enzyme complementary in the second case and the first case. So this is about activation of energy. The here the enzyme so which is having in complementary with this substrate is having less activation energy, and it gives more rate of reaction than comparing with the two states. And in this slide, so we're going to learn about the mechanism of enzyme action. So here we can learn the role of enzymes in converting the substrates into products. Here we're having four important stages. The first stage is substrate is entering to the enzyme active site, and the second stage is enzyme substrate complex. The third state is enzyme product complex, and the fourth state is the products will be relieved from the enzyme. So these are the four important stages. So while the mechanism is occurred, and here we can take two important examples. The here we are taking a lactose sugar, so which is acting as a substrate here, and which is catalyzed by the enzyme lactase. So now the lactose is a sugar, so which is converting into glucose and galactose. These are the final products by the catalysis process. And another example is that hydrogen peroxide is acting as a substrate and which will be catalyzed by a catalase enzyme. So this catalase enzyme will be catalyzing this hydrogen peroxide into water molecule and oxygen molecules. So this conversion of substrate to products will be explained by two important theories like lock and key theory and induced fit theory. So we can learn about these theories in the coming slides. So these are the two important theories. So first one is lock and key theory. And the second one is induced fit theory. In lock and key theory, so important thing is here the substrate is acting as a key and the enzyme active site which is acting as a lock. So a key fits into the lock. So this is the main concept which is involved in lock and key concept. But here the important point is the active site of enzyme will be rigid in nature according to this concept. So due to this rigid nature, so only few amount of substrate molecules will be attaching at this position, and this is not flexible. So that's why due to rigid nature here most of the times this theory will be failed. So why? Because it is not explaining about the allosteric modulations like allosteric inhibition and allosteric activation processes. So we cannot explain by this lock and key theory. So that's why this model is a failure model. And the second model is called to be a induced fit model, so which is successive model. So here important point is the active site of this enzyme will be flexible. So due to flexible active site here the substrate, so when the substrate is attaching to the active site of enzyme, so which induces, that means the substrate induces the enzyme conformational change. So that means here the shape of the active site will be changing according to the shape of the substrate. So that means the substrate is inducing the change in the conformational state of the enzymes. So that means here a specific substrate will be making or will be inducing a precise conformational change in the active site of the enzyme. So that's why here the substrate can be easily attached at this position, but initially the active site will be different. But after conformational change, the substrate will be attached at active site, and there will be a suitable chemical environment will be present in between these two substrate and active site. So here, so when it is attaching with the active site, here it is getting the highest point. So that means it is getting the transitional state. So due to getting the transitional state, here the activation energy will be reduced. And after transitional state, due to developing of some electrostatic repulsion forces, so here the products will be released and the enzyme is again getting the previous orientation, and it is involving in the further catalytic processes. So this is about induced fit model, and so this is a very successful model than comparing with the lock and key hypothesis. Why? Because here the active site is flexible in nature, so it can be suitable for most of the chemical reactions. And here we are having four important catalytic processes of enzymes. The first one is catalysis by proximity. So here the catalysis by proximity, the proximity term which is also called to be a distance or orientation. Here the substrate molecules will be oriented so very near to the enzyme molecules. So So if you take two cases, that some substrate molecules which are very close to the enzyme, so will be having more probability to attach with the enzyme at enzyme active site. So if the substrate molecules are very far from the enzyme active site, there is very less chances to attach with the enzyme active site. And here, so by maintaining the a bond forming distance with the enzyme active site, so here the concentration of substrate molecules will be increased at active site. So due to that, a correct positioning of substrate molecules will be aligned with the active site of enzyme, and so due to exactly fitting into the enzyme active site, and that decreasing the activation energy, so which leads to decrease in entropy. Here the term entropy is very much important. So why? Because the substrate molecules which are present outside the enzyme, so which are disorientated, so that means the entropy will be depending upon disorientation. If more disorientation will be present, that means it is having more entropy. Here outside substrate molecules are having more entropy when it is comparing with the substrate molecules which are present inside the active site. So why? Because inside the active site, so these substrate molecules are properly aligned or oriented, so entropy will be decreases. So due to decrease of entropy and a highly ordered enzyme substrate complex, so here the rate of reaction will be increased in this catalysis of proximity. In this catalysis of proximity, so we can also learn two important terms, that is desolvation process and electrostatic destabilization. And desolvation process, here water molecules will be removed. The water molecules which are present around the substrate molecules, as well as the water molecules which are present inside the active site. So while attaching this substrate molecule to the active site of enzyme, here the water molecules will be removed. This process is called to be desolvation. After dissolution of water molecules, here the enzyme substrate complex will be formed, and these water molecules will not be, it is not involving in the reaction. So why? Because these are dissolvated. If the water molecules are present inside the active site, so these water molecules might be reacting with the substrate and forming another compounds. So that's why this dissolution process is very essential. And the next process is called electrostatic destabilization. So here electrostatic uh, forces will be developed here. So after attaching of substrate to the enzyme active site, here there is a repulsion process will be developed, and after converting the substrate into the product, these repulsive process also will be increased, and finally the products will be separated from the enzyme active site. So this is called to be electrostatic destabilization process. This is the one of the type of catalytic process, so which is called catalysis by proximity. And the next catalysis process called to be acid base catalysis, so which is this mechanism is also called to be keto enol tautomerism. So if you see this reaction which is uncatalyzed, here there is no involvement of any enzymes. Here the ketones are converting into enols, so by reaching to the transition state. So here the H plus ions which are removed from this carbon, and after that at enol state, so H plus ions are again adding to the O carbon. That means here H plus ions are removed from this carbon and it is attached to the O oxygen molecule, and this is called keto enol tautomerism. So here this reaction will take so much time and the time consuming process. So why? Because here the enzyme is not present, it is uncatalyzed reaction. And here we are having two catalyzed reactions that are called acid catalyzed reaction and base catalyzed reaction. Acid catalyzed reaction is containing an acid, so proton donor, so which is present at the active site of enzyme, so which donates H plus ions to the oxygen molecules. So when H plus ions are donated to the oxygen molecules here, so H plus will be H plus molecule which is attached to the CH2 will be removed. So when H plus is removed from this CH2, here the double bond will be forming, and here, so OH hydroxy group will be forming at here. And A minus, so will be removed. So A minus will be present at the active site of enzyme, and after that the again water molecule is which is present at active site will be providing H plus ions, and again it is converting into Ha. So that means initially the proton donor which is present at active site of enzyme, so after involving of the reaction again it is converting into a proton donor. So this is called acid catalysis process. Here Ha is acting as a proton donor which is present at active site of enzyme, and this is the substrate. So that means H here, so acid which is providing H plus ions to the substrate. The second catalysis process is called base catalysis process. Here the base is present which is present at the active site of the enzyme, and here the base will be accepting the H plus ions from the substrate. So after H plus ions from the substrate, here the destabilization will be occurred, and here oxygen will be getting a minus charge. And after adding of water molecules from the water, again here these H plus ions will be attaching at O minus region and forming hydroxy group. So this is about a base catalysis process, and these proton donors and proton acceptors which will be present at the active site of enzymes. So these are the amino residues which are present, which are involving in the acid base catalysis. And so these amino acid residues which are present at the active site of enzyme, and here these amino acid residues which are having proton donors and proton acceptors. So for example, cysteine is having this proton donor, and this is the proton acceptor. So in that way, so all the amino acid residues are having proton donors and proton acceptors, so these will be useful for the acid base catalysis process. The third catalysis process is called to be catalysis by strain. Here the strain forces will be applicable by the enzyme. So here important point here is the strain, the catalysis by the strain is mainly occurred in few enzymes like lyases. So these lyases uh, will be acting on the substrates, and which breaks the substrate and it is converting into two products. And here important thing is the substrate, so which is not completely aligned with the enzyme active site. That means there is unfavorable conformation will be present in between enzyme and substrate. So that means there is a gap in between these two enzyme and substrate molecules, so which leads to the catalysis by the strain. So due to filling up this gap, so here the enzyme so will be which will be straining, which will be stretching the bonds, and here it is stretching the substrate molecules, and it is reaching to the transition state here. So complete binding energy will be increased, and here at this position it is reaching to the transition state, and at this position the activity, sorry, the active bond, the active energy will be decreases, the rate of reaction will be increases. And after this transition state, here the substrate molecules will be broken and it is converting into products. And this is the most rate enhancing mechanism, so comparing with the other mechanisms like catalysis by proximity and acid base catalysis and some other catalytic processes. So this is the most rate enhancing mechanism. So why? Because here the strain force will be applicable on the substrate molecules. And the last fourth catalysis process is called covalent catalysis. A covalent bond will be forming between enzyme and substrate. So this is the enzyme and this is the substrate. Here a covalent bond will be forming between enzyme and substrate. So this is called covalent intermediates. After formation of covalent intermediates at transition state, then so these products will be released finally. So substrates are finally converting into products. So here we can have one important example that is called chymotrypsin, so which is showing covalent catalysis and also acid base catalysis. So in this covalent catalysis, two important steps will be occurring. The first one is nucleophile attack of enzyme on substrate. So this is the nucleophile, so which is attached with serine. Serine is the amino acid. Here 195 is the number the amino acid number, that 195 serine is having a nucleophile, but here hydroxy molecule is present. So initially the H plus ions will be accepted by this nucleophile, so which is also present in this active site. And after H plus ions are released, so here this oxygen molecule, which is having a lone pair of electron and which is trying to form a bond with this compound, another compound. Here this is the substrate molecules. Here it is trying to attack carbon. So these electron pair, so which is given by a nucleophile to the carbon, and these electrons will be accepted by a electrophile, so which is having a plus charge. The nitrogen is having the plus charge, so which will be accepting the electrons from the carbon, and the bond will be broken in between nitrogen and carbon, and here finally amine group will be released. And these covalent catalysis process will be generally occurred in group transfer reactions. And finally, so here intermediate will be forming. So after transition state, here the bond will be broken, and finally the compound will be, the products will be released. So this is about the catalysis uh, sorry, covalent catalysis process. So here it will be occurred in between enzymes and one or more substrate molecules. So chymotrypsin is the main example for this catalysis process. So this is the end of the topic. Thank you.